Hello everyone, welcome to another neuroscience video. Today we're going to talk about the receptors of the neurochemical called glutamate. So glutamate is the major excitatory neurotransmitter of the nervous system. And it's represented in these red dots in this picture. So here we have a presynaptic neuron here that is sending glutamate to this postsynaptic cell. Now, the postsynaptic neuron expresses two types of glutamate receptors. One of them is AMPA here in yellow, and the other one's NMDA receptors. Now, what will happen is that glutamate will enter the synapse and bind to these receptors. When it binds to AMPA receptors, AMPA would open up its sodium channel, right? And so sodium would begin to come in and enter the cell through AMPA. And this would cause a depolarization of the cell. So the inside of the cell would become more positive and the outside would become more negative. If you do the same thing to an NMDA receptor as you did to the AMPA receptor, right, you would get the sodium channel of the NMDA receptor to open. However, notice what's in here. There's a magnesium ion that is blocking the NMDA receptor. So sodium cannot come in. However, if there is enough neurotransmitter being released, and it's being released frequently enough, a lot of AMPA receptors will be stimulated. So they will allow a lot more sodium into the cell. Right? And if enough sodium comes into the cell and the cell is depolarized enough for long enough, then what can happen is that the magnesium ion will be like, look at the outside of the cell. It's very negative, and I'm very positive. So I want to be on the outside. So the magnesium ion is actually going to be expelled out and go to the outside of the cell. So this magnesium ion would go away. So this magnesium ion is now it's now here. And this would open up the NMDA receptor, and the NMDA receptor would allow sodium to flow into the cell. However, it doesn't just let sodium flow into the cell, it also lets calcium flow in. And when calcium flows into the cell, this is important because calcium can act like a second messenger. Calcium is going to basically activate certain signaling cascades that are going to activate protein kinases and lead to a change in the gene expression of the cell. The outcome of that gene expression is going to be that the cell is going to come in and phosphorylate some of the AMPA receptors that it has. And when you phosphorylate an AMPA receptor, you basically increase the amount of sodium that it can let in. So at a future time point, once the NMDA receptors are activated and you get this signaling cascade and change in gene expression, at a later point when you have the same amount of glutamate binding to this receptor, you will have more sodium coming in, right? So this basically increases how much signal that this postsynaptic cell can receive from the same amount of glutamate. And also the change in gene expression can make it so that the cell is going to express more AMPA receptors. So we're adding more AMPA receptors. Right? And as neurotransmitter binds, we're going to get even more sodium into the cell. So all of these processes are basically going to enhance the synapse. And it's going to make it so that it's going to be easier for this postsynaptic cell to fire at a later point in time after this process has occurred. So this is one of the basically one of the mechanisms that's proposed to be responsible for long-term potentiation. Long-term potentiation is when a synapse is strengthened due to constant and frequent stimulation, and this is basically what happens for memory formation. The synapse gets stronger, and so you don't need as many cues in order to remember something. And in the synapse, that's represented in that you don't need as much glutamate to make this neuron fire. So I hope that helped you understand NMDA and AMPA receptors, and thank you for watching.